Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Using High-Density Electrophysiological Recordings to Investigate Neural Mechanisms in Small and Large Animals. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. This webinar has been sponsored by Diagnostic Biochips, so a big thank you to them for helping make the event possible. We're very fortunate to have Michael Long and Kari Hoffman with us today. We'll be discussing high-density electrophysiological recordings and their application to the study of the neural mechanisms of learning, memory, and behavior. I'd like to welcome Brian Jameson, founder and CTO of Diagnostic Biochips, to say a few words and also introduce our speakers. And thanks so much for joining us today, Brian.
Excellent. Thank you, Andrew. As Andrew mentioned, I'm Brian Jameson, DBC's CTO or Chief Technology Officer. I did found DBC back in 2013. And I have to say, there's no false modesty required for me to say, I'm not the smartest person here. I'm not the person who knows the most about neuroscience, not the best engineer. And on many days, I'm not even the most hardworking person here. All of those superlatives go to our great team. So specifically, Greg Alden for putting together the team in the first place, our CEO. Scott, who is on the call, our sales and customer service manager. Fawn with new product development, whom you will get to know hopefully as you buy and use our product, and then the manufacturing team. So Nick, Lee, and Grace as well, and then many others behind the scene as well. The one superlative that I can claim is I've been here the longest, and so I'm the person that sort of gets to tell our origin story, of which the full version is way too long to give here, but I will give you a very abbreviated version. Some really important characters that figure in that story. One is my thesis advisor, Ken Wise, who is arguably the inventor of neural probes and also happens to be Fawn's grand thesis advisor. Another name that was very important to our origins is Yuri Buzaki. So Yuri, very, very presciently or perceptively back in 2006, started talking to us about how neuroscience tools, specifically for circuit level neurophysiology, were really going to get a lot more attention in terms of needing improvement and becoming the focus for additional investment. And I'm glad we listened. It was Yuri's sort of connection of us to Howard Hughes Medical Institute, Genelia, that really kicked off the company. And then we also listened to his advice and ended up getting some Brain Initiative funding to do our early technology development. A third character that you may be surprised to hear about, I did a 5-year stint or sidetrack after learning this probe business at NASA and worked under a world-renowned planetary scientist there named Hasso Niemann. And what I learned there was what an incredible difference in terms of enabling new science it makes when you put engineering and R&D in service to great scientists rather than building things and then hoping someone will use it. And so when we formed the company back in 2013, it was with the mission to provide the very, very best tools for neurophysiology and to let science drive that process. So as we put it, the science is hard, the tools shouldn't be. And so today I'm very pleased to introduce as our speakers two of the people that I think most of when I think of that model. And they are Michael Long from NYU and Kari Hoffman from Vanderbilt. Both of them help us be better, and they use both the carrot and the stick at times to help us deliver on our promise. Kari does some really amazing work with chronic deep array probes in awake and behaving macaques, and she's going to talk about that work today. Michael also uses our deep array probes, but what he's going to talk about today is his use of our very small, compact, high channel silicon probes, specifically in songbirds and also chronic preps of singing mice. So without further ado, we're going to start with Michael, and over to you, Mike, and thanks for coming in today.
Thank you, Brian. This is a treat. I think working with Diagnostic Biochips has done an incredible amount for our science, and Brian and Fawn and Scott are great, and the whole team, and we couldn't do what we do without them. And so a lot of what I'm going to show you is thanks to their efforts. I've decided to do a small departure at the beginning of the talk to talk about something that's patently not silicon probes, but just to motivate why we're going towards silicon probes in that specific case. And then I'm going to talk about a really strange animal, one that we're just beginning to characterize now, and understanding how small animal silicon probes can help us understand the brain in that case. So the general theme that ties all this together is neural mechanisms of interactive communication. And I don't know if anyone has noticed, but there's been a pandemic, and a lot of our interactions are really limited to Zoom, if that, right? So I think it's been a very strange time, and I think it is, as you can see through this cartoon, leads to really Zoom fatigue, which of course is something that we can all roll our eyes at, but we can't roll our eyes at, is the fact that about 15% of our population suffers from communication disorders, everything from deafness related all the way through various stroke-related aphasias and autism. And we need to learn much more about how the brain actually communicates than we know. And so our first few slides here are just trying to look exactly at that effort. I want to point that speech, even though many of us do it quite closely, and after learning it when we're maybe 1 or 2 years old, is really complex. So words have a rapidly changing acoustic structure of on the order of 20 milliseconds. Each phoneme requires the net movement of about 100 different muscles: respiration, lips, tongue, jaw, larynx, all to make words that you can understand. And so for those 100 muscles all to work together to do anything is tough for those of us who are better at science than, say, throwing a baseball. But it's something that we all can evolve to do. And it's not only that we speak out into the openness, right? We have partners in crime when we speak often. And this is what that looks like, interactive speech. So what I've done here is just taken 2 minutes of a podcast. And this is when this person was speaking here. The width of that shape represents the passage of that person's speech. And then this other person speaks here. And what's clear is that there are moments, and this, of course, makes a lot of intuitive sense, where things just kind of bounce back and forth, kind of like hitting a tennis ball back and forth over the net. And this is known in the field of linguistics as turn-taking. And people have studied this explicitly. And turn-taking in people happens with incredible rapidity. So this lag between speakers is on average 200 milliseconds. So that is lightning fast. That's about as much time as it takes for you to blink your eyes. So how does this work? Well, a lot of psychophysical work has been done to show behaviorally that speech planning is part of this rapid back and forth. And that, of course, also makes intuitive sense. We're thinking about what we're going to say, and then there's a process of actually executing that. And we think that planning is something that can really be affected in a number of different communication disorders, but we don't really know much about how the brain does what it does to plan for interactive communication. So we decided to put together a study. This was published in January of this year in Nature. And in that study, we used these electrodes, which go right on the surface of the brain. These are electrocorticography recordings. Here, instead of doing things like what Kari is going to show us, where we can actually really get beautiful units and understand something about the computations that are being done at a single neuron or groups of neuron population level, we are hearing the roar of the crowd. So each one of these white dots here can have the ability to look at the brain with reasonably good spatial and temporal resolution, 10 millimeters and 10 milliseconds, but clearly not anything what Kari's going to show you later on with single spikes and single neurons. I should point out that the volunteers for this study were in neurosurgical theater because they're undergoing treatment for medically intractable epilepsy or brain tumors. But importantly, they could still chat. They could still talk back and forth, even with these tools touching the surface of their brain. And so this is when an experimenter is talking. This is the duration of that person's speech. And the participant can answer often with sub-second precision. Great. So what do we do to get to speech planning? You can imagine if we wanted to look at motor activity, we just simply would have the person repeat a bunch of syllables, and that's what we do to study speech production. If we wanted to look at perception, we could just play a bunch of speech and look at, for example, responses in the temporal lobe in primary and secondary auditory centers. And this is work that has been really spearheaded by Eddie Chang and others using these same kind of electrocorticography tools to get to the sensory and motor side. This is a more cognitive aspect, right? So planning, how do you actually read that person's mind and see these kind of planning processes as they bubble up? So we decided to use this really clever paradigm, clever because we didn't come up with it. It was actually developed by Bogels and Levinson. We had to translate it from the original Dutch. That was the one heavy lift that we did. It looks like this. So there's a bank of questions. These are 4 examples out of 20 questions. And each one has this kind of blue word. That blue word is a critical word. And once the person hears that word, they can start to plan their response. So how many fingers does a person have? How many toes, arms, legs? Once they hear that, they can start to plan, I have 2 legs. Right? And there's a bunch of different questions that we've used, antonyms, animal sounds, etc. And we simply could look for things that turn on with those critical words. And this is something that has been done kind of behaviorally, but we want to use these kind of high-resolution tools or moderate resolution tools to really try and figure out what the brain is doing during this computation. So this is a picture of what we can find. So what I'm going to do is just show you average responses, and the averages are going to be demarcated by this 200 millisecond shaded region, okay? And I'm going to show you an average response in a grid that is right here in this part of the brain. So this is in frontal and parietal cortex, and then there's a strip down here in auditory cortex too. And the responses are going to be shown with these circles. As they get bigger and redder, that means that there's more activity, and the activity, as we define it, is high gamma activity. So we just do this, and we do a power spectrum, and that kind of roar of the crowd that has some kind of relationship with multi-unit activity, if you like, that's been shown by a bunch of groups. So this is what happens in the 200 milliseconds before the question. You can see things are quite quiet. Then 200 milliseconds after the question starts, auditory cortex lights up. That's great. Auditory does auditory. But when that critical word happens, so you can see the shaded region going in and starting this kind of planning process, what lights up but Broca's region? As a Broca's region had been traditionally thought to be really important for the expression of speech. A lesion there leads to Broca's aphasia, which is the inability to produce fluent speech, but that appears to be really active during this planning phase. Then this migrates over. This is the 200 milliseconds before the response. You can see many electrodes lighting up here in speech sensory motor cortex. And finally, this is when the person is speaking. These actually are active in the auditory cortex due to reentrant speech. And then finally, back to zero. So just to show you that progression again, this is the time before the question. Auditory starts. There's some planning activity that's bubbling up here. That's transferred over to this speech sensory motor cortex, and then done. Which I think highlights that even though we're looking at high gamma activity, we get a pretty high resolution view. In fact, the first electrocorticography view for this kind of interactive speech. Okay, so what did we find? We can revisit all 8 of these patients, and again, about half of them were suffering from epilepsy, the other half had brain tumors. What's interesting is that we see the same phenomenon in both cases. So this isn't related to the underlying pathology. And what do we find? So we would expect to see some sensory responses here, as well as maybe in the frontal aspect of the cortex. Indeed, we see exactly that. We would expect to see motor responses, primarily in this primary motor and somatosensory cortex, and that's where most of them are restricted. But it's anybody's guess on where we may see those planning responses, and people have proposed a number of different places. And what we find is something that seems to be a hot spot here in frontal cortex primarily. I'm referring to this as an interactive language hub. And so another way of looking at the data is like this. And so the percentage of active electrodes is shown with this kind of hot spot here, primarily in this middle frontal gyrus and in Broca's region, as we saw in that one patient in the movie. So one question is, that's fantastic. What about in these less scripted tasks? When we're getting together with our friends, we don't necessarily talk about cows that moo, or what's the opposite of this and that, or how many toes do we have? What about natural conversation? So this is what that looks like. And so we can actually look at individual electrodes that gave us motor responses and planning responses in that more rigid task, and ask what happens when those electrodes are still on the brain and we're talking to the patient about what they had at cafeteria, did they watch the Dolphins game yesterday, this and that. So we asked, were you born in Mexico? New Mexico. And we said, have you been to Mexico? No. Never? Never. So what you can see here are clear red bumps, and this was in an electrode that we had previously characterized to be a motor electrode. And indeed, when the person speaks, we see these motor responses. But we also see these blue bumps here. And that, we think, is, in this ethologically relevant back and forth, clearly a window inside that person as they're planning their response. When we say, were you born in Mexico, you can see these things kind of bubble up and then finally get executed at the level of speech motor cortex in a kind of alternating fashion. And the final thing that I want to show with the human story is, we see planning activity in this hub that's primarily in the frontal cortex. We wanted to ask if this is necessary for normal vocal interactions. And one way we could do that is actually pass a small amount of current at the times that planning is supposed to happen. So this is an electrode where we saw that same kind of planning response. And we can pass current in half the trials. The current is shown with these horizontal yellow lines. That's the timing of the current. These are all those critical information questions. So the question is here with critical information in blue, and the participant's timing, so the reaction time, is shown in red. And you can see this kind of histogram here. They're a little bit slower than average, about 400 milliseconds. And that gets significantly slower when we stimulate. So there's a clear kind of slowing down of this snappy back and forth. But what I think is really much more compelling is that we start to see these errors. So in about an order of magnitude more cases during stim, we see errors like this. Say the plural of man clearly into the microphone. The answer, of course, is men. They say man. They turn it into a repetition problem, right, as we're stimulating here. So there's something where we can actually stimulate and cause an error in the lexical retrieval aspect of planning. Which word is the opposite of fast? Stimulation here, rapid. So they can still speak. I want to actually really point that out. If we stimulated, for example, primary motor areas, then they feel a kind of sense that their tongue is frozen and they don't produce speech, but that's not the case here. The opposite will be slow then. So this brings us to a device that we're working with Brian and Fawn with, and that is to try and understand what's going on with the resolution that can be achieved in other non-human animals. And that's by using this we call the bee stinger, and it's something that has really high density electrodes and go into the brain. This is actually us testing this in a songbird. So this is this part of that bee stinger is lowered into a nucleus in the songbird called RA, and we can see for 32 electrodes that are in that part of the brain, we get 33 identified neurons, and they're firing away like this. And recently we've been trying this in human participants, and this is a case where over 100 identified units have been found in this human participant. We're still tweaking some of the details, and it's still very much an experimental device, but it's one where we can, I think, break free from the kind of high gamma shackles and understand what's going on at a microcircuit level. So in this part of the talk, I want to say that speech perception, planning, and production are separately represented in the human neocortex. There's this language planning network for interactive speech. It includes these regions, middle frontal cortex and Broca's region. And consistent cortical responses occur during scripted tasks and natural conversation. And perturbation of this activity leads to slowed interactions and linguistic errors. And I wanted to take a much smaller amount of time and talk actually about small animals, because it's the title of what I'm supposed to be doing today. And I wanted to motivate it with this. So, traditionally people have used an entire zoo of animals to discover things, and went back to look at 38 of the animals that have been used to make Nobel Prize discoveries since 1903. These are the top 20 in terms of the frequency that they're used. And this is how those 20 are represented in a recent SFN abstract. So you can see this is the Society for Neuroscience. The chips have all been really placed on a small number of animals. And that tide, I think, is starting to turn. There's a lot of really bright people out there that are looking, for example, at Yale, Elena Grichova and the 13-line ground squirrel, octopuses, poison frogs, chickadees, mouse lemurs, and many other creatures are starting to bubble up. And the beauty of what Brian and his team can do is develop tools that allow us to really break free from just the mouse. And we can put those high-density arrays in any kind of creature. And so this is one of the creatures that we choose to put our array in, and that's the singing mouse. So the singing mouse is native to the cloud forests of Costa Rica. They have a very clear back and forth behavior. I want to say that Steve Phelps was the one who really characterized the singing mouse first in an evolutionary biology perspective. We work with a number of other folks to help analyze our data and to collect our data. And this is what that looks like. So those are two singing mice in very different cages in one room, singing to each other in an interaction that's referred to as counter singing. I want to first take you through the structure of that song. So this is looking at frequency and time. It's a sonogram. You can see that they make these kind of downsweep notes, and the notes get a bit longer in time and then kind of die out, lose steam by the end. But that's what their song looks like. And the charm of these animals is that they actually sing with each other, right? So there's a kind of reaching out in the same way that we reach out in conversation. And so here's a case where this white singing mouse, which was living in the room for several months, is interacting with an intruder mouse. This is a red mouse. That red mouse has an answer for this white mouse. No matter what he says, he'll start a conversation and end it, or he'll just end it when the first mouse starts. And what's interesting is if you look at the timing of that and we align to that resident mouse in white, you can see that mouse in red happens with really fast precision with respect to his vocal partner. And so if we look at human speech, remember I told you about 200 milliseconds, here it's about 500 milliseconds. And so we see this kind of distribution like this. And the leading model for interactive vocalizations at this point has been the marmoset, which is a small primate. And we've learned a ton from the marmoset, and in no way putting it down, but you can see that these interactions are much slower. So you can imagine sitting around a room and chatting and giving 5 seconds in between every response. I think we'd all go mad. So I wanted to wrap this up with one little result here about using the diagnostic biochips to record activity, and that is we found this hotspot in the brain. We could stimulate different parts of the brain, and when we stimulate it here in this orofacial motor cortex, it leads to very robust flexion of vocal musculature. And so we put electrodes in that area, and we can record, in this case, 28 simultaneous units. We've done better since then, but the reason I wanted to show these, this is our first data set in the singing mouse. And these guys are singing. There's a lot of engineering kind of constraints here. They're moving, kind of almost seizing during their song, and we can hold these with implanted animals for weeks and weeks. And it's really a beautiful tool for this job. During singing, you can see the activity of these 28 neurons seems to evolve in a very similar way. So each line is a cell, and you can see that cell changing its frequency in some cases in an orderly way across all songs. We don't see that if we look outside of singing. Also, if we look at the songs themselves, this is in shaded here is one cell when the animal is producing song. You can also see that same thing where the frequency is shown as a color plot, and it's heterogeneous, right? So sometimes there's blue before the onset of the song, and sometimes there's yellow, so high firing. And so we can organize those and actually split off the times when the animal is singing by himself. So this is the beginning and the end of the song, and you can see there's very little activity here. And times when the animal is singing in response to a partner. So there seems to be some kind of detonation that's happening here, some social modulation. And that's something we're very excited to follow up in these creatures. And the final experiment here is we can actually get these guys to sing with a loudspeaker. So they have counter singing with this kind of playback of their conspecific song. And the playbacks are shown over this hour-and-a-half period as a vertical white tick. And then these dots here are the times when the animal responds to that playback. you can see if we just inject saline into the brain, which is meant to be a negative control, there's very faithful responses to playback. If we inject mucimol, which temporarily inactivates that small portion of the brain, the orofacial motor cortex, we see the responses fall down to almost zero. But luckily, there are some responses left, and what we see is something that really resembles actually that marmoset timescale, something that's 5 to 10 seconds. And we think that this is probably driven by other circuitry, something related to arousal, likely anterior cingulate, and we're looking into this now. But the fast back and forth is gone. And so what this means is that in both cases, the humans as well as the singing mouse, there's some kind of sensory trigger, a critical word here, some part of the mouse's song, that leads to motor preparation in this interactive language hub or maybe orofacial motor cortex, and then very fast responses. And if we manipulate that and we cause disruption with stimulation here, it leads to slow interactions and linguistic errors. And if we inactivate in the singing mouse, it abolishes that interactive countersinging. And that's really the story I wanted to tell. And thank you all for your attention, and happy to take any questions.
Thanks very much, Michael, for the wonderful presentation. I think Brian will introduce our next speaker here, Kari, and we'll get to Q&A after her presentation.
Sure, sounds great. Thank you, Andrew. So, and thanks, Mike, for a great talk, Michael. So from humans, birds, singing mice, we move swiftly on to a story about macaques. And Kari's prep has been something that we've been working on with her. And I say prep because there's been a lot of engineering that Kari has done to make this prep work in a chronic basis in freely moving macaques. It's a pretty remarkable set of experiments, and Fawn and others here have been doing their best to keep up with Kari and her requirements for mechanical packaging and putting multiple probes in single animals. And so she is really the chief engineer on this project, but as I indicated earlier, she's really set the bar very high for us in terms of trying to accommodate these really elegant experiments. And this is a work in progress, as you'll hear, with more surgeries planned in the near future. So without further ado, over to you, Kari.
Thank you so much, Brian, and thanks, Michael, for setting the stage here. And also, I should say, for kind of making this possible, it was the Long Lab that did some of the first recordings with these deep probes that he let Brian and Fawn at Diagnostic Biochips show me the data. The physiologist's equivalent is show me the money. And that's what convinced me that it was worth giving them a shot, specifically these deep probes. And so we haven't looked back. So thank you, Michael Lunn, for being generous. And we're hopefully in this session going to pay it forward a little. As Brian indicated, it's not that we are extensively far along. And so I won't be presenting rich, detailed, quantitative statistical results, but we've had enough luck with them that it felt like what it would mean to pay it forward would include something like this webinar. And to give you an idea of the impressions we have from the work we've done on them so far, I also want to give a shout out to John Wolfe at Penn, who we have been collaborating with on some of the technical aspects of this development. And he's been a source of encouragement as well as a great resource for the electrode development. So thanks, John. And finally, the folks in the lab who have been doing the day-to-day difficult work of doing the recordings. So Sam Abispour is a PhD student that all of this work is going towards his dissertation. And Ken Raman is joining the lab with his master's in data science, and he's an incoming PhD student. And I'll show you some movies at the end that he was instrumental in putting together that includes some deep lab cut video tracking from the wireless data. So again, Sam and Ken have been absolutely indispensable in this process and actually are right now probably doing a recording session from our second animal. So to get rolling, the reason that we're interested in high density linear probes, I'll come back to these points with some results slides, but I want to touch on them. If you have similar interests that already, these might be some keywords for you to say that what we're looking at might also be applicable to your application. So microcircuit computations are of interest to us. We want to understand when we say, neural circuit mechanisms of, in our case, learning and memory, part of the computations we're interested in mean resolving the microcircuit by looking at different spatial inputs. So laminar resolution, being able to dissect the current sources and sinks as part of the microstructure of a region and as a way of looking across regions. The ensemble unit activity. So we're interested in memory trace replay and state changes that we identify as a function of ensemble spiking units. And that means we need, in many cases, well-isolated single units. And due to the sparsity of firing, we need a lot of them. And functional cell types. So again, I'll touch on this with some slides from our results, but whereas we see some amazing advances that are coming from smaller animals by being able to use genetic tagging of specific cell types, there are some physiological characteristics that we can use with probes like these deep array probes that are allowing us to get at some of the functional cell types, even in the macaque. We're interested in local and long-range computations. So for learning and memory, there's some question about which regions are contributing when and how from the time you learn about something until you might remember it, sometimes days later, maybe weeks, or in some cases, we've even looked at responses that macaques have shown for memory that can be as much as a year or a year and a half old. And so being able to look at local and long-range computations is really important for us to try to understand how memory might change over the course of time, and specifically the representation of memories. And there's a trade-off in the resolution that you can get at being able to detail those local circuits versus being able to monitor long range. And then the final piece, which is an essential piece for our lab, is to be able to do moving animal, and that means chronic implants. So indwelling arrays that will allow us to record wirelessly. And we're interested in this partly because we think it's important to add naturalistic experimental paradigms to the field. This increases the external validity of the studies that we do. So it's the most likely to look at how the brain has evolved and how it, let's say, naturally wants to respond to things. So when we talk about mechanisms, we're not looking at mechanisms as we've imposed them in a lab setting, but hopefully how they are in the real world. It also can be the case that complexity matters. And so Michael Long talked about some of the auditory and vocalization stimuli. And we know that as auditory cortex is one example, the responses you get to, say, pure tones versus complex and naturalistic tones is quite different depending on where you are in auditory cortex. And just expanding on that as you look at the natural variance that is present in the stimulus milieu in our environment, that the complexity of neural responses should be tailored to match that rather than tailored to match our univariate simple stimuli. So we think that's actually important to measure. And finally, we're interested in immersive embodied cognition. So just as with the vocal turn taking and responses that you saw, being able to get an embedded response from our animals, we think is very important. Not only to being able to measure the brain as it's kind of designed, if you will, to respond, but also because we want a very rich stimulus space, because we're interested in memory traces, and the more impoverished the input, the less likely we are to be able to track repetitions of activity-related ensembles. So that's our interest. So what's the big deal? Because all of the things that I'm listing on this page, there are existing electrodes. There are actually a variety of electrodes that exist that do that. So our issue is getting electrodes that have the constellation of properties to get us what I've just described, but in the deep structures, in the full brain for large animals. And so what you're seeing here is the 2-scale. And you don't usually see these things 2-scale because it's actually kind of hard to even visualize some elements like the mouse brain or bits and pieces within the mouse brain if you're also scaling up to a macaque brain. On the bottom, you'll see those red tick marks. So you can imagine those are all 2-scale. So a deep probe in a mouse can be 3 or 5 millimeters long. And there are a number of options to get you there. And there have been for quite a few years now. The options to be able to get you structures deep and far in the macaque are far more limited. And we've tried a number of those different approaches. So I'm going to talk to you today about why we've been having a special luck with the deep array probes. But leading up to this point, the work that I'd done in my PhD was to develop a 576 channel single channel array for recording in the macaque. And this was in the lab of Carol Barnes and Bruce McNaughton. And so this single channel high density, it was 4 arrays of 144 channels each, is a sort of a prototype to the style that you might be familiar with as the gray matter drive. And this drive that we had developed at that time was inspired by some work that was being done in Mike Mersnick's lab. So everything kind of builds on each other. This gave us single unit ensemble responses. And that was a start, but we wanted more. And as I've described before, there's some things that were afforded by adding in the spatial richness and the waveform richness from linear arrays and dense arrays that we weren't seeing with the single electrodes. And in fact, the unit isolation we demonstrated when we moved on to tetrode drives, that having the multi-channel approach allowed us to resolve units that otherwise we would have conflated. And that's a particular problem when the firing rates of units are low. And we have a lot of those types of units. So sparse firing units, it's harder to tell from, for example, short ISI distribution, whether or not you've got a well-isolated unit or not from a single channel. As you add multi-channel in, that gets better. And so as I started my lab, we were developing and using tetrode drives, whether those were twisted wire drives or Thomas recording quartz silicate. And we had some luck with those, again, with the single unit responses. They were on drives, which allowed us to lower in rays. And from that, it became really apparent that that functional localization was a key parameter for us to gain certainty about the structures we were recording in. But we didn't want to completely, give up tetrode channels for being in areas superficial to or deeper than the areas of interest. And so we started to move to linear arrays and to increasingly high density arrays. And so we've tried with flexible polymer style arrays and silicon arrays, 16 channel, 32, 64 channel, a number of different vendors, Atlas, NeuroNexus, Cambridge Neurotech. So we've tried a lot of different electrodes out and have had varying success with them. But overall, amassed, I've been using various multiple channel solutions for the last 20 years. Now I'm finally in a position to say, I think we've got something that at the moment we can move forward with in a good way. And my hope is actually that in 5 or 10 years from now, we're going to be even on to improved technology. And I think it's companies like Diagnostic Biochips that have the vision to see where the need is and to see where these things are going. For me, it's not about one company or one electrode, but that we are going to need a variety of solutions to be able to have our electrodes and our technology be working in the service of our questions and not the other way around. So what we've been doing now, most recently in the last, I guess it's been almost a year and a half, we've recorded now in 2 animals. Importantly, the first animal was a retrofit, meaning we had tried some other electrodes in a chronic implant already. And the way I do chronic implants, the real results would come from the recordings that you get after that initial sealed recording. So I have some caution. I'll try to answer your questions as faithfully as possible as I can to what our experience was. But you should know going in that the starting point was a retrofit. And so some of those aspects will make that a suboptimal outcome, I think. And we're in the process right now of recording from our second animal. We recorded from 2 styles of the Diagnostic Biochip deep array, 64 channel and 128 channel. We've used linear and bilinear staggered, which would be like the bee sting that you saw from Michael Long, and then also a kind of 3D. So as the probe itself has a 3D element to it, we've tried to capitalize on that to increase our yields. We have recorded, and here's, I think, one real key right now is we've recorded from electrodes that are 20 to 45 millimeters in length. And really, those aren't super limited. So one thing that I like very much as you're getting started with this kind of a probe is that you're not asking for some unique run or some massive tool up to be able to tailor the length to your needs. And there's no reason from 45 millimeters that it can't be longer if the approach you have requires a bit steeper on top of the head to allow you access. We've used 30 to 90 micron pitch, and I'm happy to discuss in the Q&A some questions about that. I think there is an important point, especially compared to other high density arrays, that there's going to be a trade-off between channels and density. And if you have a limited amount of channels that you're trying to divvy up, you're going to want to be judicious about decisions about the pitch and coverage versus how many probes you use. For us, we've gone up to about 5 millimeters of coverage. Again, that's going to be dictated by the channels and the pitch that we're concerned with. But it has been important, and I'll show you in the next slide what we think is interesting and important about being able to get a pretty fat or long coverage while not compromising our ability to resolve with multiple channels the single unit activity. Let's see. In terms of a little bit of longevity, and again, some of this I'll put over in the Q&A, but for our first animal, she's still implanted. It's been about 1.5 years. We've exchanged electrodes, so it's not that the electrodes are all 1.5 years, but so far there doesn't seem to be a hard limit in terms of sort of the implant or electrode longevity. Importantly, we always use drives. So when we implant, we have micro drives that allow us fine adjustment. That's super important for us, because then if we see a signal start to fade or we want to optimize, we can bring the electrode tip up out of the area of interest and then lower it again. And with that refresh process, so far we haven't yet come into a limit where that seems to have somehow run out or whatever. This resampling method seems to be working for us. We're able to get the units like what I'm going to show you for. You get some drop off in the week range, but for us, it seems to be working to hold a position for a week. And as yields maybe start to decline a bit, do a refresh. And generally speaking, it seems like when we're in a good cross section through gray matter that we're able to get good signal to noise units once again. Okay. So coming back to why we're interested in this, one feature that has me quite excited about using these probes is the input specificity in space. And so what you're looking at right now is a coronal cross-section from the macaque, and the CT co-registered to MR image of our first animal, where you can see that the electrode was placed so that it can access the CA1 of the region of the hippocampus. In this implant, we've also targeted multiple other structures. So it was a 4-electrode implant by design, and including retrosplenial cortex and some other regions of the hippocampus. But for this particular electrode, what this is allowing us, as you can hopefully see from the cross section and then the expansion in the middle is that the pyramidal cell in CA1, that red zone, is the pyramidal layer. And the inputs to CA1 have an anatomical segregation. And that's something that's conserved across species. What this is allowing us is we can look at intrinsic hippocampal signal, so signal that's coming from CA3 into a specific region of CA1 compared to those that are coming from outside the hippocampus. So neocortical inputs to CA1 have a different pathway, and it lands in a different area. Not only that, but there's a body of evidence to show that there are different frequencies of oscillations, and there are also different interneuron cell types that are contributing to the processing of these two distinct streams of signal. And we have access to that with these electrodes by looking in the distal regions of the dendrite, the proximal regions of the dendrite, and then around the cell body area. And so on the right, you can see an example where Sam's made a nice current source density plot around the sharp wave ripple. The sharp wave ripple is a feature that we see in the CA1 pyramidal layer via inputs from the stratum radiatum. And in the monkey, it also extends into the pyramidal layer. And so that's just one example, but of course, there are a number of different oscillations that we see that we can then characterize in terms of their sources and sinks to try to better understand the neural circuit functions within this area. So another thing that we've been able to assess with these electrodes, you can see some of the ensemble responses. So the peak waveforms across channels for different units are shown on the left. And then the way this would look as you divvy this up by single units is shown in the middle column. So you can see not only do we get traces of waveforms across channels, which is important because it means the single unit isolation process is not about the signal to noise on one channel. It means it's about that n-dimensional signal to noise. And so for us, that gives us much greater confidence in our isolation and the ability to isolate more cells. So on the right, you can see an example raster plot over time. So the y-axis there is going to be the different isolated single units recorded over time. Now, what I hadn't fully appreciated until we started getting the data in was the exquisite level of detail of some of the spiking characteristics we were seeing. So in addition to looking at waveforms, which can help us to identify different functional cell types, we also have spiking statistics. And because we've identified the peak waveforms, so how many channels they span and also where they exist relative to the local fields, we can start to further identify different putative cell classes. And finally, we're getting local interactions across these areas. So, for example, if you have very short latency responses across two waveforms, you can look at, for example, evidence of short-term or even long-term plasticity by comparing the input-output functions of those two. And that's not something that I thought really would be within the regime of what we could do with the awake behaving macaque. All right, so just to give you a little flavor for where this goes. And if you're interested more in the experiments, I'm happy to chat more about it, but I just want to finish up to give you an idea of where we've gone with this. We have an enclosure that allows our monkeys to explore 8 different touchscreens, 4 touchscreens per side of this enclosure. And I'm going to show you a movie that basically gives you what you need to know about this multisensory object and scene experience. So I'll show you here. If you look in the lower corner, you can see the deep lab cut tract response of the animal as they're going to make a Z-shaped pattern across these touchscreens. Now that's the goal. This is a task that requires the structures we're recording from. And you can see in the lower right the LFP, it's been subsampled because there's so many channels, you can't really fit it in or appreciate it if you don't. And you can see we also have the spiking activity. And what this is then giving us is the experience of the animal moment to moment. So we can start to collect up what are the consistent responses we see as the animal makes this journey repeatedly. And we can ask if that kind of activity is replayed in rest in a way that hadn't occurred before the experience. So this kind of reactivation is what I looked at in my PhD. And people are also interested in humans as we talk about reinstatement. Because we have population activity, we can look moment to moment for these blips when the brain state or neural ensemble changes. It might change from the local representation to, say, what we would consider a memory trace, something that might predict before retrieval. So for us, this is a very important and interesting space that was made possible or requires the ensemble activity. And so I'll finish up showing a little bit of what we mean when we talk about that state space. So from the population's response across multiple single units, you can imagine that you get this n-dimensional vector of responses that if you were to repeat something and the cells were to respond exactly the same way, that n-dimensional point would be very close to that replay or that next n-dimensional population. And as things change, they will become more and more distant in space. So you might also know this as representational similarity analysis. So using this kind of an approach, you can ask about different ways the brain might be representing the animal's trajectory, the animal's experience. And this might vary across areas as well. And so we're now in a position to compare this neural population activity from one versus another area as we look at events that are familiar to the animal from the recent past or from the remote past. Finally, when we apply that similarity analysis, it allows us to compare differences across screens, so is spatial content important? Is it the journey? What's happening in rest beforehand? What's happening in rest afterwards? So these are just three different ways that people can depict these population spaces. But I would emphasize that for us, this doesn't have to be a pseudo population where you're averaging across trials. Instead, we can look for those moment to moment hiccups or transitions where you see the brain is occupying a look ahead space or a reminiscence from a past activity space. So for us, that's a very important avenue that we are interested in looking in, that we think these chips are actually allowing us to do. Okay, so I'm happy to address the sort of conditions that were the most important for us in developing the technique to be able to obtain the results that I showed you. And I want to give another shout out to Fan Wu and Brian Jameson in particular at Diagnostic Biochips for allowing me to wield not just the carrot, but occasionally the stick to get this to happen. Thank you.
Thank you both, Kari and Michael, for the wonderful presentations today. Those were both fantastic. I really enjoyed them. And with that, let's jump right onto the Q&A. So everyone's back with us. Welcome, everyone. Let's kick off here, perhaps, with a question about the use of the probes in zebra finches. Have you been able to use these probes in zebra finches? You indicated that these probes can be used in any animal, and it seems that finches would have complications that another type of mouse, for example, might not, such as flight and skull thickness. So perhaps, Michael, maybe you could tackle that one first.
Sure. So thanks for the question. We have indeed used these in zebra finches. There's a paper that came out in December last year, and I can actually forward that to Andrew, and he can. People are interested in the details of that. And these were absolutely DBC probes that were made to be lightweight, so that they were minimally invasive for the birds. They sing happily within tens of minutes of an implant, and it's true that if they sing, they're happy, and unless they sing, we don't get data. So the beauty about having something that's so light and so streamlined and so friendly to small animals, it really made our job easy.
Wonderful. I'll just note for anyone, we are going a little bit over time here. If you do have to leave us, thank you for joining us. The webinar and all the resources will be available on demand shortly. And thank you very much for joining us. Moving on to another question. Thanks for the great response, Michael. I suppose this is for everyone. How do you handle noise in recording resulting from animal movement? It's always an issue.
I'll jump in with the wireless. The noise sources can be different, but one of the things that we've done with these probes is using different reference and ground selection channels. And again, shout out to John Wolf, who has a similar setup. And then we've been using for that reference, so connecting to the intracranial for the reference part, and then skull screw for ground. Fortunately, like knock on wood, our wireless recordings with the Neuralink's billing system are surprisingly low noise. And we sometimes find that the extra equipment involved in our rig setup with the N10 sometimes even noisier than the wireless recordings actually. So yeah, I guess answers separating out reference and ground can be helpful, and having a deep reference can also be helpful in our hands.
Wonderful. Did you have anything to add to that, Michael, about stability?
No.
Okay, great. Thanks very much, Kari. We do have a question about implantation techniques. I'm not sure if we have the time to get into that. Perhaps a future webinar. If any of you have any comments about the peculiarities of implantation very briefly.
So why don't I do it quick, and then Michael does the other extremes. So I would just say that we were pleasantly surprised. We can penetrate fresh dura with the monkey without any extra dural puncture or dissection or incision. And so we just use the electrodes there to penetrate dura, and then we seal it with a silicone, different grades of silicone elastomer, to make sure that it's sealed after implant. Michael, I don't know if you wanted to add.
Yeah, different animals have different challenges, but that's roughly the way that we do it too. Yeah, very well put.
Great. Thank you both. A question. How was your experience recording animals in a semi-natural environment, and were there any unusual neural patterns? And are you planning to compare activity from restrained and freely moving animals? I think that may be directed at you, Kari, but I'm not 100% sure.
Oh, I assumed it was Michael.
Perhaps,
okay.
Yeah.
So in the freely moving compared to restrained, we haven't seen as many differences as I expected. What we thought was the theta oscillation, which is so prevalent in the hippocampus of the rodent, would suddenly appear in the macaque, and it didn't. So seems to be the case in humans and macaques that regardless of walking around or free movement or not, that the theta oscillation is something that happens as the animal's sleepy, and that it's much more likely to see high frequencies when they're moving. So it's kind of a negative result, but it was an important one because we needed to actually measure both restrained and moving conditions to recognize that difference.
Great. Did you have anything to add to that, Michael? Sorry to put you on the spot.
Yeah, no, thank you. We, again, a happy animal is a vocalizing animal. And so I think, in terms of restraint, different experiments call for different things. But for the most part, they're hopping around their environment, flying, singing, doing whatever. And I think I was very interested to hear Kari's view on this, because I think there's been some controversy in the Campbell field about what people have done up to this point, which is using really high density electrodes and putting the animal in a virtual environment. And so basically bringing the environment to them in a kind of closed loop put them on a wheel and have that move the environment as if they're playing a video game, if you like. And there has not been an enormous amount of agreement, at least from my kind of vantage point, which is not in the front row like Kari is, but concerning whether or not that actually recapitulates what's going on in the natural, navigating animal. And clearly, one thing that is a problem with head-fixed animals is the fact that they're not getting any vestibular inputs, right? So that's something that I think is really exciting about Kari's work, is that not only visual flow, but vestibular inputs and all other environmental cues. It's really exciting what she's doing. It's a treat to hear about it.
Great, thank you both. Great answers. I think we'll make this last. There's two questions I think we can clump together here. I think we'll make them the last, just in the interests of time. The first question is, what is your opinion on rigid probes? Do you notice any brain damage due to the rigidness of the probe? And another question I think is related. Have you checked the stability of the recording, and have you established whether you recorded the same neurons over time? So I suppose tied together in terms of electrode placement after the experiment, and also any damage due to placement.
I'll answer quickly and then give way to Kari's answer, which I think is going to be much more relevant. I would say, have colleagues up at Columbia in the Axel lab that are recording for 2 months, and they've really shown, I think beyond a shadow of a doubt, that they are recording the same neurons for the period that exceeds 28 days, for example. And this was a paper that came out last year in Nature, Schoonover, Fink, and Axel, worth looking at. I think in terms of stiffness, we were actually talking about this exact issue right before this event, and that is, there's something about being floppy that is really forgiving, and the brain bounces around all the time, and but that same floppiness makes it hard to really enter the brain in a clear way. And so we were trying to imagine, a way that could actually hit a button and adjust stiff going in, and then allow it to, be more forgiving once it's actually in the brain. So if anyone of you 55 that are still around have fantastic ideas, I'm sure Kari and I would be all ears. This is relevant as of an hour and 7 minutes ago. Kari?
Yes. In principle, that is where things need to go. Somehow we need to have the maximal compliance with brain tissue. In practice, I've yet to see deep probes, and I've used a number of them, including flexible ones, that can get the yields over time. And so we need to be able to drive after the fact. We need the ability to adjust afterward. And that adjustment means some degree of rigidity. So there's a compromise in having electrodes that are fine enough that they're still somewhat bendy, but they're drivable. And right now, in my assessment, that's the sweet spot for being able to get units and yields that are fairly stable over time, over days, if not weeks, and continued responses count on recognizing that ultimately will do less damage, and we have hopefully potentially better yields if we could manage to get the truly flexible. In the meantime, I'll be recording my population responses from
Okay, wonderful. Unfortunately, we are a bit over on time. I think we'll have to leave it there. But I'd just like to say thank you to Kari, Michael, and Brian for sharing all your wonderful insights with us today. And it's been an absolute pleasure having you with us.
Thank you.
Thanks, guys. Fantastic.
Thanks.
So just before we leave here, I would just like to say a big thank you to the audience for joining us, and particularly as we've gone over time and participating. And last but not least, we'd certainly like to thank our sponsor, Diagnostic Biochips. So in closing, we hope you enjoyed this Inside Scientific webinar, and we hope to see you again with us next time.